start with this. Same thing is cell membrane system. Okay, so you have to remember this and the structure of cell membrane: lipid bilayers, transport protein, and integral protein, and phospholipid is there, then movement between these inside the cell, and it is also in the cytoplasm. Now lipid constituent about approximately 50% of mass of most of the cell membrane, and strength the properties varies depending on the types of the membranes. Okay, so I am repeating same things, what is their composition and that. Okay, phospholipid, phospholipid are predominantly membrane lipids in the both prokaryotes and eukaryotes, require both membrane structure and function. They are fatty acid ester characterized by the presence of phosphate residue and depending on the nature of the alcohols. Residue is present in that. Okay, phosphatidyl is inositol, phosphatidyl choline and sphingomyelin, that is cephalin, that I have already been taught to you. So here is the phosphoglycerides. Phosphoglyceride is a phosphatidic acid where phosphate residue is located, as I said in previously, that is carbon 3 of diglycerides, and C1 is generally saturated fatty acid with the C2 bears unsaturated fatty acid, that is that may be a C18 to C20, and the presence of phosphate group make a phosphatidide and chiral highly charged polar compounds. And second, alcoholic groups or hydroxyl groups of phosphatic residues can be esterified with the another highly polar alcohol moieties, that is gives you a phosphatidyl derivatives in the most common phosphatidyl glycerides. This substituent groups of, as I said, phosphatidyl choline, phosphatidyl ethylamine, phosphatidyl serine and inositol, these are there. A phosphodiester bond, thus like a glycerides, a backbone with the substituent groups. And note then, any given phosphoglycerides actually represent a molecular species in the membrane, which have a same or all structure but differ in their specific fatty acid constituent, that as I said. Okay, so you have to remember these names and their functions and where it is attached. Now the plasma membrane. Okay, you have to understand these two. These plasma membrane, for example, are approximately 50% lipid and 50% protein. The inner membrane of mitochondria, on the other hand, contain are usually high fractions, about 70% of the protein, reflecting the abundance of the protein complex involved in the electron transport and oxidative phosphorylation. Abundant of the protein complex which are involved in the electron transport and oxidative phosphorylation. Though plasma membrane have a membrane channel proteins and peripheral protein and receptor proteins and certain carbohydrate motifs is there, then glycocalyx that is found in also plasma membrane, the external membrane surface and phospholipid bilayer. So these are the various differences and where the cholesterol is synthesized and where it is located. So understand these, you have to remember what is in plasma membrane. Now I am telling you how it is moved and what is the principle behind that, and how the membrane transport is possible. Okay, so the membrane transport raw materials are enters the cell. Water, that is water exiting the cell in continuous flow or traffic across the cell membrane. The cell membrane can regulate this flow of traffic by being a selective permeable membranes. This selective permeable membrane through which the molecule is transferred. So one of the best example, imbibition, you understand, concentration. Okay, same water molecules will differentially permeable to the membrane system, and sometimes sucrose having a higher molecules and that has concentration is also higher than the water molecules. So the movement is like this. Okay, depending upon the concentrations in the higher to low or low to higher. Now these membranes, how this transport is possible and what are the principles behind that? So the movement of substances across the plasma membrane or a membrane structure, that need for the movement of substances across the membrane. Sometimes it is also useful that there are four responsible factors or types that movement of the substances will decide. That may be through a passive transport or active transport. So principle behind active transport and passive transport I will give you later on, but you have to understand what is the need for the movement of substances across the membrane. To obtain the substances require cells. This is the major function too. Two objective behind that. Needs for movement. Okay, across the membrane. To eliminate substance, that is waste product from the cells, which are not useful for the cells, that should be out from their environment. Same is to absorb a certain required molecules in the membrane system. Now I am giving you a passive transport. There are three features. One is simple diffusion, another is through osmosis or through facilitated diffusions. Why this is osmosis? Osmosis, you know, yes, effect of different concentration of the solution, okay, that will move in the animal and plant cells, higher concentration to lower concentration. There are different applications too, but this is the movement substances across the plasma membrane, and you have to write one or two sentences from this, okay. So draw like this or remember like this, so you can write very well. Now I will continue with this. In this slide you have to remember two terminology. One is phagocytosis, another is pinocytosis. What is phagocytosis and what is pinocytosis? That you have to remember. Now, phagocytosis, pinocytosis, that is also movement. The movement of solid particle in a cell through the formation of membrane vesicles. Structure you see. I am repeating this. Phagocytosis means the movement of solid particles into a cell through the formation of membrane vesicles. Same thing is, here you have to remember pinocytosis. The movement of fluid droplets. I am repeating, movement of fluid droplets into a cell through the formation of non-membrane vesicles. Right or wrong? Okay. Here is also membrane vesicles. Here is also a membrane vesicle. You have to remember only a movement of solid particles and movement of fluid droplets. Okay. So what is phagocytosis? That is solid. And what is pinocytosis? That is droplets. Okay. Remember this. Now another is yes or no is substituent moving towards the membrane system. It also facilitates the diffusion, the movement of substances down, okay, through the membrane droplets or through the membrane systems. Simple movement or simple diffusion, the movement of substances down concentration gradients without the use of, yes. Now what is osmosis? As I said in previous, this slide will not show you what is written. So what I am speaking, that you have to remember. Okay. So I am repeating this. The movement of proteins. No. Here is movement of water through selective permeable membranes from a size of high water concentration to the or solid concentration through an area of low water concentration. High solid concentration layer. This is the moment. Okay. Osmosis, as I say, higher concentration to lower concentration. This is the moment, only a simple moment. Diffusion, you know, this is the structure here that shows you a diffusion. Okay. So there are a movement, as I say, transport movement that depends on these three, that is osmosis, then diffusion, passive transport. Okay. Now I am giving these secondary active transport, exocytosis, then primary active transport, ATP is used. Here is ATP used directly. Okay. This is the phosphatidylipid bilayer, through how this happening through the cell. Okay. Then here is the machinery that will show you food particles and proteins and fluid droplets, whether it is big one or smaller one, how it is living and entering in the various system. So that is already been taught to you in the above slide. So I will not go in depth for that. But to understand, if you are not understand in previous slide, I will give you a different language and different understanding. Movement, that is simple diffusion, facilitated diffusion and osmosis. Water molecules diffuse across the selective permeable membrane from the area of higher concentration to the area of lower concentration, that is called osmosis. Now facilitated diffusions, if you see the description of facilitated diffusion, first is carrier transport protein in the plasma membrane temporarily bind with the molecules and help them pass across the membrane. No cell energy, simple diffusion, no cell energy is needed. So molecules move down concentration gradient, molecules spread out randomly from the area of higher concentration to area of lower concentration until they are distributed evenly in the state of dynamic equilibrium. That is simple diffusion. So remember this osmosis and simple diffusion, that is not much more differences, but here in osmosis you required a selective permeable membrane systems. Okay. Whereas in simple diffusion, that molecules move down concentration gradients. Molecules spread out random from the area of higher concentration to area of lower concentration until they are distributed evenly in the state of dynamic equilibrium. So this is the simple diffusion and movement across the. Now properties. Before the understanding of these, you have to understand what is the properties of plasma membranes. So we will study it in the next lecture. Thank you.